Welcome to the Deep Dive, where we cut through the noise to get to the core of what truly matters. Today, we're plunging into a fascinating frontier, the world of RNA research and advanced data analysis. Imagine a hidden language spoken by every cell in your body, dictating life and death, health and disease. For decades, it was, well, a whisper, too complex to fully decipher. Our mission for this deep dive is to explore how groundbreaking approaches are finally giving us the Rosetta Stone to this cellular language, unlocking secrets that could rewrite medicine itself. We're not just reading messages, we're trying to understand the very intent behind them, shaping everything from cancer therapy to drug development.
It's definitely an exciting time because biological discovery is now so deeply intertwined with sophisticated computational power. We're moving beyond just, reading the genome. We're just like having a static blueprint.
Right, the blueprint.
Exactly. To truly understanding the active dynamic processes of life. The answer, as you suggest, lies in the nuanced world of RNA, the real-time orchestrator of cellular activity. This integration of biology and data science is really how we extract truly meaningful insights from increasingly complex data sets. It's accelerating both understanding and innovation.
Okay, so if we're talking about deciphering the cell's real-time instructions, what are the most pressing problems this specialized RNA research and advanced data analysis are helping to solve, in biotech and pharma?
Yeah, think of it this way. We're not just doing general science. We're helping to potentially cut years off drug development by pinpointing crucial disease mechanisms. We're also dramatically improving early cancer detection and even unlocking entirely new therapeutic targets that were, well, previously invisible to researchers.
Invisible, wow.
That's the real impact of specialized RNA analysis. We collaborate with pharmaceutical, biotech, academic organizations to leverage this advanced data analysis and drive breakthroughs in critical areas like cancer research and therapy, also accelerating drug development through preclinical screening, enhancing liquid biopsy and diagnostics, and pushing the boundaries of target discovery.
That's a compelling shift from what could be happening to what is. You mentioned translatome-driven data as a core area. For those of us maybe more familiar with traditional RNA-seq, how does focusing on the translatome and techniques like ribosome profiling or tRNA sequencing offer a fundamentally different or more powerful lens?
That's a great question. The key here is perspective. Instead of just looking at the potential for gene expression, the translatome, that's all the messenger RNAs currently being translated into proteins, gives us a real-time snapshot, a snapshot of what a cell is actually doing right now, not just what it could do.
Okay, so it's like moving from a script to a live performance.
Precisely. A live performance. Our expertise lies in precisely handling these complex data sets. We process over 1,000 samples annually, so we're geared up for this. The goal is to deliver accurate, meaningful insights that answer even the most challenging biological questions. And this includes those specialized areas like tRNA sequencing, ribosome profiling, and our advanced RNA-seq services.
This sounds like where your true differentiation lies then. It's not just the biological expertise, but how you integrate it with custom software. Could you elaborate on that, how that combination creates something, well, unique in this field?
Absolutely, yeah, this is what truly sets us apart. We recognized early on that the power of this data needed to be accessible. So we developed a proprietary Python-based platform called Martian.
Martian, okay.
Yeah, and it's built to plug right into our existing technologies, giving you this incredibly intuitive, interactive interface. The goal is really to make complex data accessible to everyone, regardless of their bioinformatics background. It empowers clients to accelerate drug discovery, biomarker identification, and their understanding of disease mechanisms through interactive dashboards.
So researchers can actually play with the data themselves.
Exactly. And some of its core modules are even published in peer-reviewed journals, which, speaks to the scientific rigor and validation behind its development.
That kind of integration sounds like it streamlines the entire research process. Let's dive into some of the specific technologies you're using. Ribosome profiling, or RiboSeq, sounds like a particularly powerful tool. What makes it so impactful, especially with these high-throughput solutions you mentioned?
Right. What's truly exciting here is our collaboration with scientists to establish a new high-throughput ribosome profiling solution. It works on a 96-well plate format. We call it Ribo 96, and it makes RiboSeq incredibly accessible and scalable.
Ribo 96, so 96 samples at once.
That's right. Before Ribo 96, RiboSeq was often quite laborious,
Right.
very low throughput. Now, we can perform parallel dose time testing, profiling multiple drug concentrations, treatment combinations, or time points in a single experiment.
Wow, okay.
This offers comprehensive condition coverage for really complex experimental designs with automated precision. And perhaps most importantly, it provides cost-effective scaling through batch processing. That significantly lowers the cost per condition, opening the door for large-scale screens that simply weren't feasible before.
Efficiency aside, the scalability of Rabo 96 and the massive data sets it generates, I imagine that's where the MLAI potential really kicks in. What specific kind of breakthroughs do you foresee when you start feeding this rich, real-time cellular activity data into machine learning models?
Yeah, that immediately begs the question, what else can we do with this kind of data, right? The large data sets generated by platforms like Rabo 96 are perfect for training machine learning models. We can use them to predict translational control elements, essentially, teaching an AI to understand the subtle nuances of how cells regulate protein production.
Okay.
But beyond that, RealWSEC is an ideal tool for detecting what we call non-canonical ORFs, or NCORFs.
And for our listeners, maybe break that down. What exactly are NCORFs and why is finding them so significant?
Sure. Think of it this way. The genome has instructions, but not all of them are officially on the map. NCORFs are like hidden messages, unannotated open reading frames that often go undetected by traditional methods. And this capability is crucial for two, let's say, sweet spots in biological discovery that are truly paradigm shifting.
Okay, sweet spots. What's the first one?
First is microprotein discovery. RiboSec is excellent for identifying microproteins. These are peptides under 100 amino acids derived from small ORFs or SORFs.
Standing proteins.
Exactly. Tiny, often overlooked, but they are critical players in areas like cancer, metabolism, immunity. Our platform can detect translation happening in what were thought to be non-coding regions, and even from non-standard start signals, non-AUG start codons. This leads to novel, disease-relevant targets that traditional proteomics completely miss.
So things other methods just wouldn't see.
Correct. And the Rabo 96 platform's scalability is particularly impactful here. It allows high-throughput screening of these SRFs across 96 conditions, even in really low-input samples like organoids or rare cells. This really is like finding a whole new set of biological actors.
It truly is like discovering a new layer of biological instruction. Given the complexity, though, what's been the biggest hurdle in translating these newly found hidden instructions into, say, actionable therapeutic strategies? And how are you working to overcome that?
Yeah, that's a key challenge. The biggest hurdle has often been the sheer volume of data and the difficulty of discerning functional significance from, well, noise. Our integrated software platform, Martian, helps overcome this. It not only identifies these microproteins, but also provides interactive tools to explore their potential roles and validate them much more rapidly. By generating these large, high-quality proteogenomics databases, we can train powerful machine learning models to predict SOR function. It helps make sense of the new data landscape much faster.
That makes perfect sense. Okay, so microprotein discovery is one sweet spot. What's the second area where RiboSec is proving to be a game changer?
The second area is immunopeptidomics and neoantigen discovery. This is incredibly important for cancer immunotherapy.
Right, immunotherapy. Big focus there.
It's huge focus. RiboSec expands the landscape for T-cell recognition by enabling the discovery of non-canonical antigens. These are derived from those NCORS we talked about, and even from circular RNAs. Recent studies have confirmed that this provides a much more comprehensive view of the antigenic landscape, what the immune system can actually see, which is directly applicable to profiling tumor versus normal samples for personalized therapies.
Personalized therapies, okay.
And our Rabo 96 platform supports this work through its high-throughput capability. We can profile, say, 96 tumor versus normal samples for these novel peptides. Its low input compatibility also makes it suitable for analyzing precious biopsies to support truly personalized therapies.
That's critical for biopsies.
Absolutely. And again, the resulting large data sets are also perfect for training machine learning models to predict which peptides will bind to MHC molecules, accelerating the search for effective immunotherapies.
RiboSec clearly opens up immense possibilities. But to truly cover the landscape of RNA research, I know you offer a suite of other specialized services. Could you briefly highlight how some of these, like maybe DART RNA-seq or nano tRNA-seq, complement RiboSec or address unique research questions?
Absolutely. Yeah, while RiboSec tells us what's actively being made, other techniques are equally vital for different insights. Take our DART RNA-seq, for instance. It's pretty revolutionary for liquid biopsy.
Liquid biopsies, okay.
Less invasive.
Much less invasive. It's designed to extract crucial insights from tiny, fragmented RNA found in blood or other fluids. This enables early disease detection and non-invasive monitoring, really a potential game changer for diagnostics. And it's currently validated for human, mouse, and monkey samples, so it has broad applicability.
Very useful. And nano-tRNA-seq. Then there's tRNA sequencing, which we call nano-tRNA-seq. Transfer RNAs, tRNAs, they are critical but often overlooked regulators of gene expression. Nano-tRNA-seq gives us an unprecedented look at their abundance and modification patterns. This offers insights into fundamental cellular processes and disease mechanisms that other RNA analyses simply can't provide. And of course, we also offer comprehensive RNA-seq analysis and translation efficiency profiling as well.
And you even mentioned something earlier that brings both RNA and protein analysis together for that kind of holistic view.
Yes, exactly. Our Haribo system, it allows for simultaneous RNA and protein analysis from the same sample. This provides a truly comprehensive view of cellular activity at that moment.
So you see the message and the result.
Precisely. This integration is key to understanding the full biological picture, as it directly links the genetic constructions to the functional output of the cell. Beyond these, we also offer custom analytical pipelines for exploring things like long, non-coding RNAs, which are increasingly recognized for their regulatory roles.
Oh.
And we can process data from virtually any species, provided a reference genome, transcriptome, and annotation file are available. This ensures broad applicability for researchers working across diverse biological systems.
This all sounds incredibly powerful, but let's talk practicalities. For researchers and organizations, how do you actually work with partners? What kind of support do you offer to ensure these complex insights are truly accessible and actionable?
Yeah, the broader implication here for our partners is really flexibility and comprehensive support. Throughout the entire research journey, we offer end-to-end solutions that include both the sequencing and the data analysis.
Okay, the full package.
The full package. Alternatively, if you prefer to handle the initial lab work, you can use our kits on your own bench and then just send us the libraries for sequencing and that integrated data analysis. We even accept externally generated RNA sequencing data. We can provide customized analysis workflows based on your specific project requirements. So really, no matter how the data is generated, we can help derive insights and push the research forward.
That flexibility is definitely key. I can see that. And once the analysis is done, what does a partner actually receive? What kind of deliverables ensure they get actionable insights, without being overwhelmed by just a flood of raw data?
Right, data overload is a real concern. All our analysis packages include an interactive HTML report. This features key metrics and visualizations, along with all relevant raw outputs and tabular files suitable for your own downstream analysis if you want to dig deeper.
It's reactive. Okay. So you can explore it.
Exactly. So for a RiboSec project, you'd get the foundational metrics like alignment quality, sure. But then we dive into more impactful visualizations. Things like metagene profiles that show you translation patterns across genes, differential analysis results, highlighting what's significantly changing between your samples, and gene ontology annotations to help you understand the biological pathways involved. Essentially, it's a roadmap of what's happening.
A roadmap. I like that.
And for nano-tRNA-seq, reports contain sequencing metrics, PCA results, heat maps, differential abundance analyses for both nuclear and mitochondrial tRNAs, giving you a clear picture of how tRNAs are responding in your system.
So it's not just data, but a guided tour through that data, tailored to provide clear insights. What is support for understanding those complex results? Translating those maps into meaningful discoveries?
Exactly. That brings up a critical point.
Yeah.
How do you translate data into discovery? We offer results interpretation sessions as part of our standard service package.
Oh, okay.
Yeah.
That's helpful.
Yeah, these consultations help clarify report content, walk you through the visualizations, and guide downstream analysis. And additional scientific support can be provided upon request for more in-depth discussions. We're here to help. We also retain raw and intermediate data securely for a limited period with long-term storage solutions available, so you always have access to your valuable data.
And typical timelines.
Our standard turnaround time for data analysis is typically 2 to 4 weeks from data reception. Now, customized projects or large-scale studies, generally over 30 samples, they may take additional time. But in those cases, we assign a dedicated project manager and a senior scientist to ensure personalized oversight and timely delivery. It's really about empowering researchers to focus on the science while we handle the data complexity, making cutting-edge RNA research a practical reality for more organizations.
What an incredible deep dive into the microscopic world of RNA and the powerful analytical tools bringing its secrets to light. From detecting those tiny microproteins to discovering new neoantigens for cancer therapy, it seems like we're truly gaining fluency in the cell's own language. The potential is truly immense.
Yeah. Zooming out for a moment, it's clear that understanding the translatome provides an unparalleled real-time view into cellular function. This level of insight offers profound hope for more precise diagnostics and targeted therapies. Therapies that truly respond to the dynamic nature of disease rather than just, treating symptoms.
So, what does this all mean for you listening right now? Imagine a future where we can predict disease progression, drug response, or even identify vulnerabilities in pathogens with unprecedented accuracy, all by listening closely to the silent language of our cells, the RNA. What new discoveries could you unlock if you had a clearer, more detailed map of life's most fundamental instructions, revealing the hidden messages that truly drive biology?